Oops. Hey guys, Michelle Glass here. What's up? Topic for today is gustation, sense of taste. So let us get started with our sense organ, which of course is located on the tongue. All right, now the tongue, you know, has a bunch of bumps on it. Those bumps are called lingual papillae. The term lingual means tongue, and papillae, that term is used throughout our coursework. That's talking about a finger-like projection. That AE ending is, of course, pluralized. So there are several of these projections on your tongue, as you know, and they have various functions. We will see one of the functions of the papillae is friction. So actually getting the food in the mouth and then having this rough surface to really be able to manipulate that food around inside the mouth. So think about scooping up that ice cream and getting that into your mouth. The filiform papillae are on the tip of the tongue, and they're really having that sole job of friction. Fungiform are named because they look like little mushroom buttons. The fungiform papillae are going to be important in friction, but also they do contain some taste buds, so they are going to be important in taste reception. Now the taste bud is actually going to be really the structure that's involved in taste sensation. They're located on the papillae. So papillae and taste buds are two different things. We also have foliate papillae, which primarily is involved in taste sensation, and the valate, which probably is the biggest one. It has about 100 or so taste buds, so you can see that that's really important for that papillae's function. So let us look at taste bud a little bit closer. So that's what we're looking at here. Remember, these are located as part of the structure of the papillae. So in white there, I've tried to draw the papilla, which is my singular version of that word. You're going to have this little gap called the taste pore, and that's where you're going to be able to get to the taste bud itself. Now the taste bud itself is going to be composed of special gustatory receptor cells. So now we're going to start seeing that pattern, which we'll see throughout, where we have a receptor cell synapsing onto sensory neurons. Our gustatory receptor cells, I've drawn in green. When they are mature cells, they do have those taste hairs, which are microvilli extending into that taste pore. Any taste receptors are located on those cellular extensions. Those cellular extensions are important in increasing surface area. So that's giving us more contact with the foodstuff or the taste ants and our receptors. Now notice you have kind of embedded in between your gustatory cell a purple cell, and it doesn't have those microvilli. The purple cells, let me go ahead and label those, are the transitional cell. So these are the cells that are immature. They're not quite ready to be our gustatory cells yet. Now the gustatory receptor cell is going to be replaced about every 10 days. So you have to constantly be making new cells to replace those old worn out gustatory cells. So in addition to our transitional cell, you're going to see, trying to make it bigger, you're going to see basal or stem cells as part of taste bud structure. So a basal cell or a stem cell, remember, is one that's mitotically active. So it's maintaining that ability to do mitosis. So essentially, the stem cell is producing more transitional cells. Those transitional cells will mature into gustatory cells, and so that you're constantly able to do that renewal. Notice that your mature gustatory cell is synapsing onto a sensory neuron, so that it can obviously send that signal on to the brain, okay? If we start mapping out our gustation pathway, you are starting with a taste ant binding to a receptor that's located on those gustatory cells. So you eat food and you smush it around with your tongue. And as you're starting to digest that food, those taste ants, those chemicals are in that mush, and you're squishing it into those taste pores, and then it's making contact with those microvilli of the gustatory cells. Now, in order to be able to taste something, you have to have a receptor that matches up in that kind of puzzle lock and key connection, right? And so there is a genetic component to taste. There are tastes that some humans can taste and other individuals cannot taste. There are things that taste good to some folks and really bad to others. Cilantro is an example that I've just recently learned about. I think it's delicious. Some people think it tastes like soap. So that's an example of a genetic difference. Now we have 4 what are described as primary tastes, and that would be sweet, sour, salty, and bitter. And then really umami is probably needing to be pushed up into that area as well, because we have a pretty good ability to describe this. Umami is that savory flavor that you're getting from glutamates, which are amino acids. So this is one of the tasty flavors for meat and cheeses and tomatoes. One of the reasons you cook broth, and the longer you cook it, the more savory it becomes, is because of that breakdown of those proteins, those amino acids, into those free glutamates. We also have taste receptors for water, and kind of a recently described one, oleogustus, which is unpleasant, kind of fatty acid flavor that's really associated with food that's off. So something that's not quite as fresh as it should be, you're able to recognize it there. Okay, so we're going to have our taste stamp binding to our receptors on our gustatory cells, and then this will cause excitation to our sensory neurons, which are going to become part of 3 of our cranial nerves. So we're going to see the facial nerve, the glossopharyngeal, and the vagus. Now, all of these are coming from different areas of the tongue. So that's partially why we have these 3. We don't need to be that detailed, but we do need to see that these 3 are involved. Makes sense that your vagus is involved. Remember, it's going to be that big part of the parasympathetic division. Parasympathetic division is involved in rest and digest, and it's controlling our organs in our abdominal pelvic cavity. And so, we're getting ready to swallow food and absorb nutrients, and we really need to start getting the digestive tract and the body geared up for that. These cranial nerves are going to come in to the medulla oblongata and go to the thalamus. The thalamus is our relay center. So now we're going to see that pattern with all of our remaining special senses, that these sensations are going to go to the thalamus and then be relayed on to the cerebral cortex. In the case of gustation, the processing center is called the gustatory cortex, and it's located at the insula lobe, which is a structure that we did study as part of our cerebrum in AMP1. Okay, let's talk a little bit more about our receptors. So these are also chemoreceptors, just like we had with olfaction, but our chemoreceptors in our olfactory sensation all functioned in the same way, and we'll see that there are 2 different types of chemoreceptors found in our gustatory cells. So the mechanism of how we detect sour and salty is the same. So both of these are ion channels. Now, different ions are going to bind to the different channels. So we detect sour is basically an acidic flavor, and so anything with an acid has a lot of hydrogen ion. So we are detecting sour by opening up hydrogen ion channels, and that's a moving through, depolarizing the cell, so it's a positive ion. Salty is detected by a sodium ion channel moving through a sodium ion channel, depolarizing that gustatory cell. So we're not seeing a binding here, we're just seeing that these ions are moving into those channels and depolarizing those gustatory cells. Our sweet, our bitter, and our umami flavors are all functioning using a G protein second messenger system, but they're specific. So your sweet, your sugars are not binding to the same receptor as your bitter molecules. Okay, so there's a division there, but the mechanism of action is the same. So once you have your sugar bind or your bitter molecule bind or the glutamate bind to those receptors, you're going to activate a G protein, and you should already anticipate that if you activate a G protein, you're going to trigger a second messenger. Remember, second messengers can be either cyclic AMP or calcium ions. And so then that's going to depolarize those gustatory cells and send that signal on our pathway. Okay.